Right. In front of you, I've shared this NCRT. Okay. Yesterday also, I told students that this is very important that you should be going through the NCRT again and again, right? It's very important so that to actually understand how the things are given in the chapter. Now, yesterday we were discussing about the ovules. You know, what is that ovule? It's a megasporangium. It is megasporangium. And what it is holding different structures. Like, just let us go through this once again. Okay. There are two polarities of the ovule. One is the callosal pole, another one is a micropyle region. Okay. So I will identify the callosal and the micropyle region. Callosal is something on which below it the embryo sac is located. Okay. And what is a micropyle? The region where the integuments are meeting each other. It's an opening kind of a small structure it is. We'll learn later that the pollen tube enters from this region. Okay, so this particular pole is known as a micropolar pole. This kind of an ovule, which has got micropole on the top and the callosal region below it, is known as an anatropous ovule. Okay, such type of an ovule, we use the one anatropous. Okay, so depending upon the place in which three things are located, one is the callosal end, one is the micropyle, another one is the location of the hilum also. Okay, on the basis of these three regions, okay, we define different types of ovules. NCRT has not mentioned any other part type, but they have just mentioned one type and they have said that this is a kind of an ovule that is called the anatropous. So it means that in your course, only one type of ovule is there, and that is the anatropous ovule. That is how you identify the anatropous ovule. So on the one side, you have the micropylar pole is there, micropylar end is there, that micropyle end will be having the integuments towards it. So you can see the two integuments are there. This outer integument, inner integuments are there. And all these integuments are meeting this place, and that place is known as a micropylar pole, that is known as, okay. Below the integument, you have very, very important structure, and this structure is called the nucellus, okay. Now, the nucellus is just surrounding the embryo sac that you can see. The green color structure inside the structure is an embryo sac, and it is surrounded by this whole structure that is called the nucellus. Remember that these nucellus, they are having a simple parenchymatous cell these are having, but these are actually providing a kind of a nutrition to the growing embryo. We'll see it later on that once the fertilization has happened, okay, the embryo needs certain things for growing in the initial stages. Okay, so nucellus is actually providing that kind of a growth for the embryo sac. It is surrounding the embryo sac. Okay, and inside you have this green color structure that is known as the that is called the embryo sac. Now, yesterday I also discussed about the different type of cells that are located inside the embryo sac. Okay, so what are the different type of cells that are located? Just a minute. Yes. Different type of cells, if we talk about, that are located inside the anatropous ovule. These are, we always talk about, just a minute. Yes. We always talk about, I discussed this in my last lecture also, that it is actually a structure that is eight-celled, eight nucleated and a seven-celled structure. Okay, how do we name it? It is an 8-cell and a 7 nucleated structure it is. So how do we, just I'll mention the numbers over here so that you understand. Now, what is this? Like you see this region, okay? Now this region is a callosal end. Okay, towards the callosal end, when the embryo sac starts dividing, okay, within the embryo sac, you have different cells are there. Now you can see the division happening. This type of a division now is a mitotic division. You know what is a mitotic division? In which the number of cells are equal, okay? So all these cells will be having the haploid number of cells each, every cell will be having. So this becomes 2, then becomes 4, then becomes 8 in number, okay? Now what happens after that is that this is nucleus actually. This is a multinucleated structure. This is the nucleus inside us, right? This is the nucleus. Now, what happens once they divide? So how many nucleus are there? 1, 2, 3, 4, 5, 6, 7, 8 nucleus. Now, they take their positions they take, okay? Finally, in case of a mature embryo sac, okay, towards the callosal end, you will have 3 nucleus arranged, okay? And that 3 nucleus surrounded by the cell wall and everything, we call it a cell now. So, antipodal cells are the 3 in number. Let me put this number 3 over here. Okay. Now, on the other hand, we have another structure that is called the micropylar end. Okay, towards the micropylar end, you have got again 2 structures and 3 structures are there. There are synergids are there. You can see these 2 structures. These are synergids. Let us make this 2 in number. And you have one cell that is at the background, and that is the egg cell. So I put in 1 number over here. So 3 and 2 and 1. Okay. Apart from that, in the central region of the embryo sac, okay, you have got a polar nuclei. Now, there's a speciality of a polar nuclei. This is a single cell it is, but it is having 2 nucleus inside it. So if I'm using the word cell, I'll put it only one. So how many cells are there? 3, 4, 5, 6, 7. So it is a 7-celled structure. This is very important. Every second year NCERT CBSE is asking this type of question in a board. So how the 7 cells I'm counting? It is 3 on the antiportal side. It is 2 towards the synergids, 1 the X cell. And this polar nuclei, it is having 2 nucleus inside, but it is actually a 1 cell. So it is a 7-cell structure. But what about the nucleus? Count the number of nucleus. 1, 2, 3 nucleus on this side, 4, 5, 6, 7, 8. So this is actually a 8. It is a 8 nucleated structure, it is, right? So you have 7-celled and nucleated structure it is. So that's how the mature embryo sac looks like. Okay, remember all the diagrams that is in front of you, that is in the NCERT, it is important. Practice these diagrams, never make diagrams which look like a rough diagram. Okay, in NCERT, biology is a subject which is full of diagrams, but you cannot be making every time a very rough kind of a diagram. Okay, so make sure that you're practicing the diagram and you are making it very nicely. One more thing I want to bring it to your notice, like, look at this diagram, this one, the where I'm putting it right now, okay? This is the diagram also of an embryo sac, and this one is also of an embryo. Both the last two diagrams are of embryo sac only, okay? The only difference between the two diagrams is that in the second diagram, last but second diagram, okay, they have not shown all the cells being made. There's some nucleus is there and few cells are there, right? So if examination, if the question comes to make a mature embryo sac, and you are making this diagram, that's wrong, right? So for a mature embryo sac, you need to make this last diagram where all the cells are nicely placed, everything is located finally, and you have that 8 nucleated and a 7-cell structure, okay? So make sure about this thing, that when you're drawing the diagrams, you should not be making mistake, because you feel that you have done something right. But when examiner checks your copies, then they understand that nobody, this thing has been not been asked in the examination, okay? So once again, I'm repeating, the last two diagrams are of the embryo sac only, okay? But in one diagram, this is not a mature embryo sac. The last diagram, this one, is actually a mature embryo sac, which you need to draw the diagram, okay? So let us proceed further from here. So this was the structure of a megasporangium. What is a megasporangium? We talk about the megaspore mother cells and all those things below that. Okay, now let us come to one again, a very important thing, because we are talking about, let me open this thing, whiteboard. Yes. So, as we are talking about a pre-fertilization event, yesterday I told you all that whole chapter is divided into three basic parts, okay? One is a pre-fertilization event, then you have a fertilization, and then you have a post-fertilization. The second part of the pre-fertilization is very important part, and we talk about a pollination. Pollination is something which you have learned in the lower classes also, I think, okay? I think they introduced these topics from the 7th, 8th standard. Now, this pollination means what? This concept is very simple, okay? The concept says that the pollen grains from one flower or some other different flower is landing on the stigma of the another flower, okay? That is the process of a pollination. Now, pollination cannot happen on its own, okay? There has to be two different types of pollination. We all again know this thing. We talk about a self kind of a pollination, right? And we talk about a cross pollination also we talk about. What are the difference between a self and a cross? If it is happening within the same flower, okay, we say it's a self-pollination it is. Okay, this is a self-pollination. And if it is happening between the different flowers of a different plants, we say that it is a cross-pollination, that is, okay. Now, in case of a self and a cross-pollination, see, if I talk about a self-pollination, obviously, the pollens are just jumping from maybe one flower to another flower, or within the same flower of a same plant, they are moving, okay. They might not be requiring any external agent for it. Okay? But if I'm talking about a cross pollination, now this is very important, especially in the case of the angiosperms, that once this pollen is formed, actually, once this pollen is formed, this pollen need to reach the top of the stigma so that the fertilization process can be initiated. Okay. Now, therefore, because pollens, they're not the motile structure, they cannot move on their own. They require some kind of agents for the locomotion, right? This is now important. These are the external agents we are talking about, okay? Basically, in the case of a cross-pollination where the pollen needs to jump from one flower to another flower of a different plant, okay? you require some kind of a agent that can transfer these pollen grains. Okay. Now, depending upon what kind of, you can say, what kind of a pollination or what kind of agents are available for the plants. Okay, we talk about different types of different type of agents. Like for example, I'll use certain terms over here. Like we use the term anemophily. I'll just write the terms, then just explain it to you, right? One is an anemophily. Okay. Then you have one more term is there when we talk about, like in case of the entomophily. Entomophily. I'll discuss what these things are. We have anemophily, we have entomophily, we have ornithophily. Ornithophily, right? And we have last one, uh, proper name is not given, but it is basically the mammal, but we'll come to it later on, okay? So when we talk about all these things, what is this we are talking about? We are talking about different kind of agents we are talking about, which are helping in the pollination process, okay? So it can be done maybe like to the mammals also, right? The mammals help in the pollination, like you and me, okay? We eat something and what we do is that we throw away the seed outside, and then seed is being used up somewhere. Maybe it's for the germination process, or maybe some animal is grazing in a forest, okay, and with them they are carrying the pollen grains. Also, that's also a possibility. So that is being done most of the mammals. Now ornithophilia, what is that ornithophilia? If that pollination is happening to the birds, right? They're small birds sitting on a flower. They're taking up the pollen over there. Like a best example of the ornithophile is a hummingbird. Many of you must have known that what is this hummingbird? A very small, little, beautiful bird. It is, okay. What does this bird do? It just sits on the flower. Okay, it continuously flies, and when it is sitting on the flower, it is actually feeding on the nectar. And when it is feeding on the nectar, at the same time, the bird's beaks and everything is carrying the pollen also. So the hummingbirds are the example of the ornithophyly where they are helping in the pollination process. Okay. Then you have entomophily, right? Entomophily is something where we talk about the insects. When the insects are acting as an agent for the pollination process, we say that it's an entomophily. The best example you can take maybe of the bees, right? The bees are happening, bees are doing something like that, butterflies are there, okay? So bees, butterflies, these are all helping in the process of pollination, and bees and the butterfly when helping in the pollination, what are they actually doing? They are actually along doing the same work. They're shifting the pollen from one flower to another flower. That is how the cross pollination is happening. Okay. Then one more thing is there when we talk about is a one more kind of an agent we talk about, a very common agent is anemophily also. You know, anemophily is a kind of abiotic agent. Okay. And in this we talk about in case of a wind, right? The wind is acting as an agent for the pollination. It is carrying the pollen grains with it. Most of the grasses like maize, okay, maize have got a very light kind of a pollen. Small wings are also present on the pollen. They just being carried away from one place to another through the wind. The very best example of the anemophile in which a wind is acting as an agent of pollination is the maize. Okay. One more I want to discuss over here is hydrophily also, right? Now, what is that hydrophily? When we talk about term as a hydro, obviously, we are talking about water, right? So in case of a hydrophily, we talk about water acting as an agent. The best example, again, we can take care of a hydrolas, vallisneria. These are the plants. Vallisneria, again, the examples of whatever examples are there in NCRT, also make sure that you are referring to these examples, very, very important examples. These are, so you have a vallisneria are there, you have a hydrilla are there, okay, hydrilla, vallisneria. These are the structures which are always using water as an agent for the pollination, okay. Most of the time, these questions come as an MCQ also. Most of the time, they are one marker question also. Sometimes they just are the definition part also. Whatever they are asking in terms of a pollination, make sure that you're always right. If somebody is asking what is ornithophyly, okay, write down it's a pollination happening with the help of the birds, okay. At the same time, write an example of, even if the question doesn't ask you the example, always mention an example because that completes your answer, okay. Entomophily, what it is happening to the insects. Again, give an example of the bee and the butterfly. Anemophily, wind. Always remember the example of the grasses like maize we talk about. Okay, hydrophiles में Vallisnerias, hydrollas. So whenever you're putting the definitions also, I would like to suggest to you that always put an example also. Okay, that completes your answer, right? So these are different type of pollination events that are happening. Okay. Um, just let me switch over my page. Okay, so as we are talking about the pollinations, I talked about different types of pollination also, what are the different types and what are the examples of the pollination. There's certain diagrams also given in the NCRT. We come to the next very important aspect. Once the pollen grains have reached, you can say, the stigma region, what happens after that, okay? So this diagram, which is A and B diagram in front of you, they're just showing you the growth of the pollen tubes, okay? How it is happening and why it is happening. Let us come to that concept now, okay? So if you look at this diagram, okay, this is a diagram which shows you this is the stigma region. This part is the stigma part, okay? And this stigma region is having lot many pollen grains on its surface. You can see so many pollen grains are there. And out of these few of the pollen grains, not every pollen grain, out of few of the pollen grain, there is a development of a very important structure that is called a pollen tube that is known as, okay? So what happens is that whenever the pollen falls on the stigma, now this is the stigma, okay? And some pollen has come in this region and it has fallen over there. For example, let us talk about pollination where the wind is happening. It's happening to the wind. Okay, wind will carry away all the pollen grains, and whenever the wind settles down, it decreases its speed. The pollen falls on the stigma. Okay, at certain place. Now there are chances that the pollen is of some different variety of a flower, and the stigma is of some different variety. Okay, so there needs to be some kind of a compatibility test between the pollen and the pistil, right? There has to be some kind of an interaction happening. This is a very important concept where we say that it is actually a pollen pistil interaction that, listen very carefully to what term I'm using. Whenever the pollen lands on the stigma, okay, there are chances that pollen is landing on a stigma of not the same species of a flower, okay, because there's no way in which that can be detected. So pollen falls on any flower. So now when it has fallen on some stigma, maybe that stigma is not belonging to the species to which that pollen is belonging, okay? So there has to be some kind of, you can say a very, if I use a simple term, there has to be some kind of a chemical reaction. Chemical identification reaction needs to be there between the pollen and the pistil, okay? And that is going to decide whether this pollen tube will grow or not. Now, this comes under the category of a pollen pistil interaction, okay? That actually is required for a compatibility test of the pollen. What does that mean? That the stigma is trying to recognize that pollen, right? Once the stigma recognizes the pollen, then the growth of the pollen tube start. And that growth of a pollen tube, you can see it happens in this way, that it grows down like that. It grows, it reaches the surface, it goes towards the other end of the embryo sac, and it enters the embryo sac through this, not the whole pollen tube is entering, it is only nucleus. And this pollen tube is actually carrying the male nuclei inside it, okay? So this is very important. When the pollination has happened, when the pollen has landed on the stigma, there has to be a pollen pistil interaction has to be there. Once the pollen passes that interaction, okay, then we come to the concept that the pollen tube starts growing. Okay, so again, now you can look at this diagram number C I'm talking about. It says that is a longitudinal section of a flower that shows the growth of a pollen tube, right? So again, this kind of a diagram also can come as a 3 marker in examination. So what is happening over here now? The pollen tube is growing, and this pollen tube grows once the you can say the pollen pistil interaction is done. Okay, now what happens next? If the pollen pistil interaction is done, okay, and next step will be obviously the male gametes will move into the pollen tubes, okay, and they are going to uh, they are going to do the process of fertilization. Let me just fine. So what is happening is that after the pollen crystal interaction, the pollen grains are now said to be compatible, okay? And once they are said to be compatible, I just made a bit larger diagram. so that you actually understand what's happening inside it. Okay, this is the stigma region, style region, and this is the ovary part inside which you have an embryo sac is located. Now you know how many cells are there in the embryo sac. You have 3 cells in this region, right? You have 2 synergids in this region. Behind you have got an egg cell in this region, and in between you are having this structure. Okay, so this is something that is there. Now what happens when this pollen grain is landed over here? Okay, now this is the pollen grain, right? It has landed over in this structure. What is the structure? The structure is this is the pollen, and this is on landed on a stigma. Once the compatibility test is done, okay, what happens to the pollen? I'll show you in a different diagram over here. This is a pollen, suppose, right? It has got a structure like this. It has got a male gamete inside. This is a male gamete. Now, this pollen bursts over here from a germ pore region. It bursts and it produces a tube-like structure, this pollen grain. And this pollen tube just keeps on growing in this region. It keeps on growing in this region. It grows, and it enters from this region. Not the whole pollen tube. I'll tell you what's exactly happening. Again, I'll just make this diagram complete over here. So what's happening is that the pollen grain, after the pollen pistil interaction, it is going to develop this pollen tube, right? Once again, I'm writing, this is the calazaline, I'm writing C for it. And this is the micropylarine, I'm writing M for it, okay? So it is entering to the micropyle region, okay? And in the micropyle region, there's a now this thing has happened. So what is the next step? It is a fertilization process which is going to happen. What is inside the pollen tube? The pollen tube is carrying 2 male gametes. Remember, inside the pollen tube, there are 2 male gametes are there, okay? So I'll just put it like this. There is a male gamete, right? And because it's a gamete, it is a product of meiosis process, okay? So it is going to have haploid number of chromosome. So you have N and N over here. These 2 male gametes are entering. Now, when this male gametes enter the pollen tube, there is a process that is happening. Now, this is an egg cell. I'll use another color for the egg cell. Just order this one. I'm talking about this cell. This is an egg cell. So what is going to happen is that once the male gamete has entered, the egg cell, which is inside the embryo sac, again having n number of chromosome, okay? Then you have one, it is going to add up with a male gamete, right? Again having how many number of chromosomes? It is having n number of chromosomes, right? And these two together are going to do the fertilization process and form the first cell of the next generation, that is a zygote, right? So n plus n becomes what? 2n. And this is the process that is known as fertilization. Fertilization. There is one more term we use for such type of a fertilization. This type of fertilization is also known as syngamy. This is also known as syngamy, right? So what has happened is that we have the male gametes having n and n number of chromosomes through the pollen tube. They have entered the embryo sac region, okay? Embryo sac region. And this embryo, just a minute, I think, this embryo sac is actually having these this was having male gamete inside it, okay? Right. So this is known as a fertilization process. Another name given to this type of fertilization is called a syngamy. Now remember when I told you that when the pollen tube is having 2 male gametes inside it, okay? Now the second male gamete, again I'll show you, is also entering this embryo sac region, okay? What is happening to the second male gamete? Let us write over here. This is the second male gamete I'm talking about. This is second male gamete, which is traveling in a pollen tube, okay? This one is also having n number of chromosomes inside it, okay? What happens to it, okay? This one actually fuses very interestingly with another cell, which is nothing but a polar nuclear, which was a double nucleated structure. If you all remember, a single cell, but a double nucleated structure. It is going to combine with a polar nuclei, right? And this polar nuclei is having how many nucleus? Two nucleus. I write it not 2n, but I write n plus n because both the nucleus are haploid in nature. So once again, the second male gamete fuses with the polar nuclei, which is already having two nucleus inside it. So what is the product? In second case, the product is a structure very, very important that is called endosperm, right? And this endosperm is a triploid structure, three end structure, two from the polar nuclei, one from the male gamete. Again, this is also fertilization process. We use another term for it. We say that it is a triple fusion. It is because triploid cell is being created. So we say that it is a triple fusion process. What is the product of the triple fusion? The product of the triple fusion is a endosperm. Got my point? This triple fusion is forming an endosperm. Now you can understand, I'll repeat all the things once again. The pollen tube after the pollen pistil interaction, after the compatibility test, okay, it grows inside the style and stigma. It reaches the other side of the micropyle region carrying the two male gametes inside it. It liberates these male gametes inside the embryosac region, okay? Now, once it is liberated inside the embryosac region, it is guarded by different type of filiform apparatus are present inside it, okay? And what are they guiding these male gametes to? Number 1 point, the male gamete, I'll write 1 and 2 words so that you understand very clearly what I'm talking about. This is the first I'm talking about, and this is the second. So what's happening to the first male gamete? The first male gamete fuses with the egg cell, okay, and forms a zygote that is a diploid structure. That is a fertilization only, okay? This fertilization is named as a syngamy. Remember the term syngamy, okay? One more gay male gamete is traveling in the pollen tube region. The second male gamete is again meeting again meeting a polar nuclei, okay? Now this polar nuclei is having two structures n plus n. The n plus n plus n, it becomes a triple fusion. It forms a triploid structure that is an endosperm. Now very interestingly, once the fertilization is happening is a syngamy, the second fertilization happening is a triple fusion. That is from where the word double fertilization comes. What does that mean? Angiosperms are having very, very specific type of fertilization process where there are two different types of fertilization. Just a minute. Fertilization, right? So the second type of fertilization that is happening, this fertilization that is happening, okay, this fertilization is, why am we using this word over here? Because it's a combination of a syngamy and the endosperm, right? I think I made the things very clear to you where this term is coming, syngamy. Whenever asked question is being asked, what is double? It generally comes as a 3-marker question. Do whatever I've done on this board. Okay, write down all the answers, all the gametes, write down, give their haploid numbers. Okay, show what the product of these fertilization process and give name to them. Draw the diagram. That's how you complete the 3-marker question. Okay, so you have an X cell and a male gamete forming a zygote. You have a triple fusion in which two polar nuclei plus a male gamete. It is forming an endosperm. And because two different types of fertilizations are happening, we use the term double fertilization. The product of the first fertilization is a zygote. The product of the second fertilization is an endosperm. Okay. Now the question comes that in the angiosperms, why such type of fertilizations are happening? What is the significance of it? Remember, every time, whatever processes are there, significance, whatever processes are happening, make sure that you are actually knowing the significance of the process. That's very important. Okay. Angiosperms may are the, are the only plants in which actually double fertilization is happening. So why is it happening? Okay. So what was the product of the double fertilization? On one side, it was forming a zygote, right? On the other side, it was forming an endosperm it was forming. On one side, it was forming a zygote. On the other side, it was forming an endosperm. Significance kya hai is cheez ka? Now, what is this endosperm is that endosperm actually provides nutrition to the growing embryo. That's why it needs to be formed actually before the formation of the zygote. If you talk about the fertilization of the two fertilization process, which fertilization is being earlier? It is actually the triple fusion. triple fusion is done earlier, and later on it's a syngamy that's happening, okay? Because even before the zygote formation happens, the endosperm is present over there. And what is this endosperm doing? It is providing nutrition to the growing zygote. Till the time the zygote becomes a seed, germinates, it forms its radicals and the plumule and start getting nutrition from the outside world. Till that time, endosperm has a responsibility of giving nutrition to the growing zygote. Okay, so this is the significance of this is how the angiosperm plants are more successful also. Okay, they're more successful higher plant these are because they have brought up with the condition that okay, the condition is that before even the zygote formation is happening, they are making this endosperm. And what is this endosperm doing? It is providing nutrition to this zygote. Okay, so when before the formation of the zygote, endosperm is there, until the time the dispersal of the seed happens and all those things are happening. We'll come to it in detail. Okay, actually, it is the endosperm that is providing food to the growing zygote. Okay, so I think till here the things are clear. What are we talking about right now? We are talking about the fertilization process. We talked about earlier as a pre-fertilization event. Then we talked about a double fertilization where we discussed about the fertilization event we discussed about, okay? And then we talked about what is known as a, sorry, then we talked about what is known as a, actually a fertilization process. Now let us come to the diagram that's given in front of you right now. We talked about the diagrams of a mature embryo sac earlier. Now there's a difference between that diagram and this diagram. This is actually a fertilized embryo sac diagram, right? In a fertilized embryo sac, the labelings will change, okay? You have the synergids. Now, what will happen to the synergids? Synergids will start degenerating. They are not no more required now. The zygote formation has happened. The fertilization is done. So these synergids are now not required, okay? So they are going to be now degenerating. What do you label over here? And you show it also in the way that it's getting degenerated. The membrane is actually getting faded away, okay? So you show it in that way. What will the second labeling of this thing? This is a zygote formation we talk about. So how does zygote formation happen? By the first fertilization process. This may we use the term syngamy. Okay, that is all. Again, on the other polar ends, the antipodal cells are present on the calasal end. Again, they are also getting degenerated because they don't have any significance now. What is there in the center? You have a primary endosperm nucleus, that is PEN, which is the result of a triple fusion. That is how it is having how many number of chromosomes? It is having 3n number of chromosomes. How many number of chromosomes are present over here? It is having just 2n number of chromosomes. Okay. So if I compare, if I just let me get back to my whiteboard again. Just a minute. Okay. So if I go back to my whiteboard once again, right. Okay. So what are we discussing now is that there's a change in a diagram now. Okay. I'll use another fresh whiteboard. Okay. To show you this thing. Now, once this embryo sac has got fertilized, you make it in something like that, right? And what do you do over here? You just show that the synergids are getting a bit degenerated. You show it this way. There is a zygote formation over here. So this is a degenerating this thing. What is this thing? This is a zygote that is, right? What do you label it zygote as? Because it is the result of a syngamy, it is 2n in number, right? In the center, again, you have what structure? This structure whole, it is known as PEN, that is the primary endosperm nucleus it is, okay? And this is a result of a triple fusion. Therefore, you label it as 3n. Very simple. But yes, many students make so many mistakes while doing this whole process. So make sure that when you are making this, when this is a diagram of a, this is a diagram of a fertilized embryo sac, right? Now you understood the difference between the two. One was the normal embryo sac, another one was a fertilized embryo sac. So in a fertilized embryo sac, you are having what? Zygote is present, and also you have this PEN, which is known as a primary endosperm. The rest of the cells are getting degenerated. Again, on this side also you can show that these antipodal cells are also getting degenerated, right? You need not show it in a very good way. Just show it that they are getting degenerated, right? So what has happened now is that the cells have now made a zygote. What is the role of this thing? Endosperm, again being asked many times in examination. So it is providing nutrition to the growing embryo. Fine. Now, what happens next after this? Fertilization has happened. Now we come to the next topic, which talks about a post-fertilization events. Okay. Earlier we talked about the pre-fertilization event, then we talked about the actually the fertilization event. Now we are talking about what happens next, right? So obviously the zygote which is being formed, it is going to divide now. So you can see this is a very good diagram they have given. They have shown you the diagram having one nucleus. You know, this is the mitotic division, normal mitotic division. Different type of shapes are there of the embryos, like this shape is actually of a globular embryo. Then you have a heart-shaped embryo is there. So you keep on seeing that these structures are being developed. Okay. And when these structures are being developed, different stages may, then it is slightly different in the case of a dicot plant and slightly different in case of a monocot plant. So we'll come to it in a post-fertilization event. The zygote is dividing. And what is the zygote doing? It is continuously, it is dividing to the mitotic process only. And this mitotic process is actually, you can say that, resulting in increase in the number of cells, right? And when it is being, you can see the different shapes are coming up. Like, for example, this is a globular shape. One large cell, small cells are there, and so many cells over here. Something like a heart-shaped structure is there. Like something, all these things are coming. And then finally, that embryo, when it is growing, it has started forming the radicals and the cotyledons and the plumule, right? Radical will, in future, you all know, form the roots, right? Plumule in future will form the stem part, right? So again, the growth of the zygote, growth of the embryo is happening and it is forming different type of forms. Now we come to a slight dissimilarity in the way the monocots and the dicots, they behave, okay? So in the post-fertilization events, we talk about this endosperm being formed, right? Successive divisions are happening in the endosperm. And let us talk about one very, very important uh, concept over here. Let me share again my whiteboard with you. Now, when I'm talking about the post-fertilization event and I talk about the endosperm, what's happening to the endosperm, right? So in case of the division when happening inside the zygote, what happens is that endosperm keeps on dividing, okay? But it is a division that is only creating nucleus in it. It is called karyokinesis, that is known as. That is called karyokinesis, right? Now, what is this karyokinesis? It is a division of the nucleus that is happening. So when the endosperm is dividing, it is initially not creating a cell, students, okay? It is just creating not many nucleus. The best example is of the coconut water. What coconut water you drink actually a nucleus. It is being said it is full of minerals and the vitamins and all those things. Okay. So what is that coconut water? It is just a nucleus. So because why it is watery? Because still the cells are not being made, right? So initially, when this endosperm starts dividing, it is only multinucleated structure, right? How does it form? It is a multi-nucleated structure, right? And this multi-nucleated structure can be best understood when we coconut water. When you bring a coconut home, a very fresh green coconut, it has got just water and you keep it just at home. What happens to that coconut water? After sometimes it becomes a bit fluid in nature, slightly less liquid and slightly later it becomes solid structure. What's happening? Initially, endosperm is just multinucleated, having just water-like structure. And slowly later it starts forming cells, okay? And finally you have that hard, thick coconut inside it. And that's when the cell formation has happened. So remember that endosperm initially is just a division of the nucleus that is happening. And initial phases may endosperm is just a multinucleated structure. Later on, it becomes a cellular structure also. So it is a karyokinesis that is followed by a cytokinesis. And that karyokinesis continues in all. So we say that it's a multinucleated structure. After some time, the cell starts developing, and that is how we say that it's not a cellular structure. So that happens in this. Now, what happens to the rest of the structure? I told you it's slightly different in case of a monocot and a dicot. So we'll see how it is different. Again, both the diagrams, very, very important. We talk about This is the embryo of a monocot and a dicot. I think my screen is shared. It is visible. Yeah, okay. Right, so this is a diagram of a monocot, dicot. Am I audible? It's showing from my end. Hello? Hello? Okay. Am I audible? Okay, so when we talk about both the monocots and the dicot, there is a bit dissimilarity between them of the embryos. Take just look at the structures, okay? So this above structure is of a dicot embryo it is. So look at the structure it is. Now again, the diagram comes in the examination. You need to draw the diagram. You have the two cotyledons are present over here. So you have two cotyledons are there. So you have the radicals are there. You have the plumule are there. And the radical, you know, that is going to later on form the root. So it is going to have the root cap with it, okay? So you have this is the structure when we talk about of a dicot embryo, right? Of a dicot plant where actually you can very clearly see two cotyledons are there, okay? When we come to the second part, again, this is again an important structure. It is every now and then it is being asked in examination. Make sure you are practicing also the structure very nicely. Okay, this is the structure of a monocot once. Okay, so in case of a monocot, is slightly different. Okay, there is a structure called coleoptile, right? If you now look at very carefully the structure, okay, you will find. We just erase this part first, and I'll explain you. Right. So this structure is of a scutellum, right? Scutellum is nothing but a cotyledon of a monocot. So monocot has got just one cotyledon and they give the name scutellum to it, okay? Then you have the shoot part and the root part. The radical is representing the root part. This is the shoot apex over here. The radical is covered by a structure that is called the coleorhiza. Can you see this over here? This is the radical part. It is having a root cap over here. Beyond those root cap, the cells which are located are known as a coleorhiza, they are known as, okay? And we look at the other side, look at the shoot apex, and below above the shoot apex, you are having again the cellular structure, and that is called the coleoptile, that is known as, okay? And this coleoptile and coleorhiza actually, you can say has this embryonical axis. इसको axis बोला जाता है, this is known as embryonical axis. This is known as this whole structure. This whole structure forms embryonical axis from the coleoptile to the coleorhiza. So it is a structure of a monocot. This is, once again, I'll tell you what is the difference between the two. So in case of a monocot, you will have just one cotyledon. The name given to the cotyledon of the monocot is a scutellum, right? And the name given to the cotyledon, we have the two different cotyledons in the case of the dicots. You can see it very separately, those two structures, okay? Remember again, the diagrams of the embryos are also equally very important. Revise both the diagrams very carefully, okay? And then what happens to when this embryo is grown, okay? When this embryo keeps on growing, it obviously forms a seed. And what are these seeds? These seeds are present inside the fruit. Remember, this very important thing is that with the whenever the sexual reproduction happens, okay, in the case of the angiosperm, the final thing which is formed post-fertilization is a seed. Now the seed will undergo the dispersal, it undergo the germination part, then the new plant will grow, okay? So what is the final product of a sexual reproduction? Somebody is asking you, it is a seed is the final product of the fertilization. Now, where is the seed actually? This is seed is inside the fruit. Fruit is formed by the mature ovary, a fertilized ovary or a mature ovary is going to form the fruit, okay? So the final product of the sexual reproduction is actually the seed, fine? Okay. Now, these seeds can be of two different types. We talk about as the albuminous and the non-albuminous seeds, okay? If suppose when the seed is growing, right, and it doesn't have any endosperm, I told you that what is the role of the endosperm? Endosperm is providing nutrition to the growing embryo because the embryo doesn't have any nutrition that is coming from any other external source. So embryo needs to provide, endosperm need to provide nutrition to the growing embryo. Now, when it is providing nutrition to the growing embryo, okay, it will either that nutrition will be completely used up, right? So the seed becomes a non-albuminous seed. Hope you're understanding what I'm speaking. When the endosperm is completely used by the growing embryo. right? The seed which is finally being formed is a non-albuminous or ex-albuminous seed. You look at the pea and the groundnut. It just have a seed cover, and inside the cotyledons you can see, okay? So it doesn't have any endosperm residue inside it, okay? There are certain seeds where the embryo is retained. It is retaining the endosperm like wheat, maize, barley, castor. Four examples they have given. So remember these four examples where the seeds are actually the albuminous seed. So what are the, what is a non-albuminous and albuminous seed? Along with the examples you need to understand, okay? There is one more thing that not only we talk about the endosperm, in very few of the cases like black pepper and beet they have talked about, okay? Even the nucellus is persistent. If you remember, the embryo sac was surrounded by a parenchymatous cell that was called the nucellus, okay? So in case of certain you can say plants like black pepper and beet, okay? The nucellus is also persisting in the seed. At that condition, that persistent nucellus is being given a new term, it is being given, and that is called the perisperm. So terminology is very, very important in our subject. What is non-albuminous with an example? What is albuminous seed with an example? What is perisperm with an example? To make sure you know the definitions of everything, and along with that, you're also knowing the examples of it. Okay, fine. Okay, so let me wind up this whole thing. So today we understood about the double fertilization process, the significance of the double fertilization process. Okay, what is the role of the endosperm being formed? Why the endosperm is being formed before actually the zygote? Because these questions do come: one marker, two marker, three questions, three marker questions are there. Okay, and what is how does a fertilized embryo sac? How does look like, okay? And then what happens when the seed formation has happened? So we talked about both a difference in the monocot seed and dicot seed, and how the embryos are also different, okay? I would like to wind up over here, okay?